everybody for coming to this training session. And today we want to talk, I want to discuss with you about phase 1 designs for oncology clinical trials. And this training section we usually give in the data science department every few years when we have enough new hires to give them some overviews about usual tools we used here. I organized my training session into a few sessions. First, is a background about phase 1 trial design oncology setting and what does efficiency actually means. And then we will review the 3+3 design, which was probably still the most popular design in oncology setting, and also some review the limitations of this design. And after that, I'll focus the big chunk of my trainings on the 3 classes of dose escalation design: the rule-based design, which we're most familiar with, you can tell from the names, and then the model-based design, which cover most of the Bayesian continued reassessment method. And the third method is model-assisted design. Yeah, I will get into details of all the 3 classes. And pretty much the names I mentioned here, I'll talk about them in details. After that, there will be some discussion on those expansion cohorts with a focus on multiple expansion cohort design, which getting popular lately, recent years. And a little bit things go back to talk about multiple escalation cohort designs. And later is a summary. So, before I get into the details of the trainings, there are a few things I want to, let you know early. First is about, this training is about oncology phase 1 trial design. Disease areas are very different. So the experience and designs and we talk about here might not be directly applicable to other disease areas. Cardiovascular, immunology, and inflammations, they could be very different settings. Second, like I said initially, I do not intend to make this training very technical. There will be not much time if I get into technical of all of them. I do not claim I'm expert on all of the designs. There's really a lot of them. As a matter of fact, I started to gather those knowledge about 2 years ago when I prepared this talk for ASH, American Society of Hematology, the scientific workshop. So you can see, my intention is I brought this out for discussions. As many questions, if you ask a question, I might not be answer some of them. Again, my goal is to give you an overview, equip you with an array of possible candidate designs for you to choose when the times you need to design your own phase 1 trials. Okay, so many attention discussion has been given to phase 2 or phase 3 designs because those designs are more closely related to drug approval. When you talk about phase 1 designs, people will think, phase 1 design is more straightforward and small scale. Actually, that's a misconception. Because of safety concerns and a serial enrollment, phase 1 trials often takes years to complete, especially if there's a novel agent forcing human trial involved. However, we know for pretty much all phase 2 and phase 3 designs, most of them have gone through a phase 1 trial stage. But many phase 1 trials did not make to phase 2 or phase 3. So they are actually much more phase 1 trials than phase 2, phase 3s. To prove my point, I get into clinicaltrials.gov and find the clinical trials registered between 2010 and 2019. It's 10 years period on lymphoma, leukemia, and myeloma. And here, at least number of trials are registered on the phase 1, phase 2, and phase 3. So you can see this number tells 43% of the trials registered during this 10 years period times are phase 1 trials. So it's a vast majority. And in the meanwhile, statisticians have developed many novel designs that generate many publications in statistical literature. However, based on my experience, I serve on SRC and Dana-Farber Cancer Center. The uptake of novel designs actually is rare, is slow. we're starting to see some uptakes, but probably does not match the pace of new designs that published on literature. So, whenever we talk about the designs, and we have to design serves objective. So we have to see what's the objective of phase 1 design, and phase 1 trials and our design need to meet that objective. To make it simple, phase 1 clinical trials is really just to identify a safe dose level to carry forward, especially in oncology setting, because we'll often talk about, cell killing, cell toxic. And so there are phase 1 trial is covered by 3 guiding principles: ethical, safety, ethical conduct, and efficiency. So patient safety guide study design by minimize the number of patients exposed to a serious or life-threatening adverse event. Means we don't want shooting patients on overly toxic dosage levels. Ethical conduct, that's actually is this is unique in oncology, different definition from other disease settings. Is usually if a patient and their physician, when they want to enter phase 1 trials, they have exhausted all other possible and reasonable standard therapy options, especially when we talk about forcing human trials. Then let's talk about efficiency. 3 guiding principle. Last one is efficiency. When we talk about efficiency, let's review what exactly does efficiency means. I copied this from FDA draft guideline on adaptive design clinical trials for drugs and biologics. So based on this guidelines, it says an efficient design can provide a greater chance to detect a drug effect than a less efficient design with the same sample size. So the sample size by greater chance. Alternatively, an efficient design may provide the same statistical power, but with a smaller expected sample size or shorter expected calendar time than a less efficient design. So now we get an idea about what is efficient means. Efficient means if you use the same sample size, an efficient design should have a greater power. If you have a you fix the power of the trial, then you will need a smaller sample size. So those things are actually the meaning of efficiency. Put this definition back into clinical phase 1 trials. You can see this means in phase 1 trial, we want to minimize patient exposed to subtherapeutic doses level. Then, so this way we can minimize the total sample size, shorten accrued durations. In phase 1 settings, we want to minimize the patient total sample size. Then it pretty much is determined by 2 factors. First is how many candidate dose levels you have. Of course, if you have 2 dose levels, you're very likely going to have much smaller sample size compared if you have 6 dose levels. The next one is about the statistical design, which is it's talk about escalation and de-escalation rules. So, whenever I talk about phase 1 designs, even for myself, when I start to work on clinical trials, and we use 3+3 designs because everybody loved them. Although this design was initially introduced in 1940s, it's still deeply loved by our community. I cited 3 review articles here. So you can see the first 2 articles, then from the times about before 2008, about 96 and 98% of phase 1 trials published use 3+3 designs. And the last articles focus on molecular target agent in relatively recent years, because we believe molecular target agent have very different, those toxicity profiles and even those efficiency profiles. However, you can see even for the molecular target agents, it still have 60% of phase 1 trials use 3+3 designs. although I have to see my data is a little bit old because I only get a review article up to 2010. I'm sure if I get a later reviews after 2010, then for the last 10 years, the percentage should be higher. And so I put a 3+3 design, the scheme, the algorithm here also lists escalation, de-escalation rule here. I don't think I need to get into details because this is really pretty straightforward. I think everybody knows it. And the target toxicity rate is between 1/6 to 1/3 as acceptable range. So, 3+3 design is, whenever I say pretty much popularity means reality, right? It's so popular in the literature. That means that pretty much that means that in reality, people use it. I'm sure, some of you, just like some of my collaborators, they come to ask me, why not, right? This 3+3 design is logistically simple. And, and clinician familiar. And from the physician's perspective, choosing a trial design that is well understood and easy to implement is of utmost importance. Because it's really straightforward to implement, like I showed in the previous slides, you just do accordingly. And for so many years, it seems to work well. And again, some of my collaborators says, this way I don't have to have a statistician. So, but I have to say this is a misconception. Although I don't want to get into details about this why, because whenever you have something not go exactly as you expected, you need to troubleshoot. For example, the passive cohort size change. You accrue 3 patients and end up only 2 become eligible. Then how do you handle those situations? So, that things you really need a statistician to come in to tell you what acceptable modification of your trials. Okay, so but of course, the reasons we talk about 3+3 designs, it has limitations. There are various statistical simulations. People have done simulations in statistical literature. Compare 3+3 designs with the hypothetical true scenarios. It finds out that it's more likely to underestimate the true MTD level, maximum tolerated dose levels, and when there are more than a few dose levels. Also, it's less efficient than due to the conservativeness and the slow dose escalation. Pretty much you start from dose level 1. Every dose levels, it will require 3 or 6 patients until it slow escalate up. So it potentially put more patients treated on the subtherapeutic dose levels. Recall what we talked about before about efficiency. When you treat more patients on a subtherapeutic dose level, means below MTD levels, then you require more patients and longer study duration. But I have to say, I have to point out here is what those limitations appears when they are more than a few dose levels. That's why I highlight them in red. When they are only just a few dose levels, if you have only 2 or 3 dose levels, 3+3 is perfectly efficient compared with any of other trials, and it's very easy to implement. So in that scenarios, 3+3 is still your top choice. Okay, I'm just, if anybody have a question, you can feel free to interrupt because I want to make this training as a discussion. Okay, so then, when I talk, okay, 3+3 have limitations, they have this kind of, potential problems. Then I guess some of my other physician collaborators come to see, what about adaptive designs? Nowadays, everybody talk about adaptive. It just sounds so cool. So, okay, before we talk about 3+3 was adaptive designs, phase 1 designs, let's go review the concept, the definition of adaptive design. as illustrated in this picture. So for the traditional fixed sample design, right? You design a trial, it's written in a protocol, and you conduct the trial, and then you do the analysis. For adaptive design, you design the trial, you started to, during the study conduct, you will review the data observed on the study so far. Then you adapt. What does adapt means? Make changes and make plans to the study. Then you continue. Then you do this loop. Then you until you reach the final analysis. Sorry. So with that being said, if you recall the phase 1 trials, for phase 1 trials, we almost always need to review the DLT and toxicity data observed on patient has been treated on the trials. Then we make a decision what to do next. So in that setting, pretty much almost all phase 1 designs are adaptive designs, although some of them were recognized as, because I think because they have the name adaptive in their name, like based on adaptive defining designs. So actually for phase 1 designs, we're ahead of other phases because we always adapt. Then, okay, so then how we know phase 3+3 designs, the histories, the background. So I want to, next part of the training is focus on the dose escalation design, currently available dose escalation designs. I quite like this classification of those escalation designs. We put all designs into 3 classes. First one is a rule-based design, which we know we're most familiar, 3+ design, rolling 6 design, and accelerated titration design. And the second class is model-based designs, then covers the continuous reassessment method and based on logic regression models. It's all pretty much based on continuous reassessment method using parametric models. And this all belongs to this model-based design class. The last class is model-assisted designs. This is a relatively new class. And there are examples like Bayesian optimal interval design, modified toxicity probability designs, and keyboard designs. Okay, so let's review the rule-based design again. So rule-based designs, we know it's straightforward and it's relatively simple. We don't have any prior assumption about those toxicity curves. So it's just every dose level treat the same, starting from the lower dose levels. So it's a no model, simple algorithms. And also this dose escalation and de-escalation rule are pre-specified. They can tabulate before the onset of the trials. You just need to do accordingly. 3 patients, no toxicity, escalate, 1 toxicity, 1 DLTs, 3 more patients. So just, as a simple algorithm can be itemized in like probably 3 lines. And when you determine MTD, when you determine dose escalation and de-escalation, only toxicity observed on the current dose level are used. So pretty much when you are done with lower dose levels, then they become irrelevant because you will never go back to them. You will not, once you escalate, usually you're only gonna de-escalate once to the potential MTD level. You will not de-escalate. You will not go up, back. And MTD level only determined pretty much the 2 levels top 2 levels become relevant. And for this class of design is logistically simple and straightforward to implement. So we talk about briefly about 3+3 designs, and this is a rolling 6 designs. Many of protocols are reviewed for oncology design. They still use rolling 6 designs. The idea of rolling 6 design, I'm sure you guys know as well, is extension of 3+3 design. The idea is just remember for 3+3 design, for after every 3 patients, we have to wait until the for the DLT observation period, usually it's 1 cycle. Once during this period of time, we cannot put more patients on the trials until we know how the DLT data look like. And this design just allow enrolling patient while we're waiting for toxicity data for patient has been treated. So it allows about 3 to 6 patients treated per cohort. The idea is to shorten, minimize the time the trial will be closed. Just no, we don't want to kill the trial momentum. We want to minimize the trial close to accrue, waiting for toxicity monitoring. And the table here on the right give you also tabulate escalation, de-escalation rules for here list number of patients treated on the trials and all possible scenarios. How many DLTs has been observed? People has been proved, patient proved with no DLT, or pending means they are still under observation. We don't know yet. And here, this is a decision rule. S stands for stay, D, de-escalation, E, escalation. Here is, there's another column that MTD exceed or not. Here means if you already you move to a MTD level, a higher dose level, which determined to be too toxic, and you move back to one lower dose level, whether that happens. So this is pretty much affect this decision here. How you do you want to keep escalate or stay? So this is the escalation, de-escalation rule for the rolling 6 design. And this is accelerated titration design was proposed by Reassessment in 1997. So this design, the idea just try to overcome the conservativeness and slow dose escalation for the initial dose levels in 3+3 design. The idea is, at the beginning, we treat one patient as a cohort, and if no DLT, no any toxicity concerning, then we escalate to next dose levels using the 1 patient per cohort until at those levels we start to see toxicity of concerning, either moderate or any sign of moderate or concerning toxicities. Then the trial switch back into 3+3 design. So this design was proposed but was never really used, getting popular. I think because maybe once we see the initial dose escalation with 1 patient per cohort, the clinician getting a little bit panic about this aggressiveness of early dose levels escalation. However, I included this design because I think this idea of the early aggressive dose escalation has some inspired the ideas in the Bayesian design later we talked. And although we, for the Bayesian continuous assessment design later, we don't know exactly, we cannot see the picture like this one, but it does allow dose escalation after the 1 patient cohort. So I think this idea probably being inspired by this ATD design. Okay.
Can I just ask a question about the rolling 6 design? So essentially, you may over enroll 3. This might, just to get to your efficiency statements, this might involve the over enrollment of 3 patients. If there's 2 DLTs, and you might have seen that only among 3.
You're right. Yeah, you're right. So this design, of course, it try to overcome, it try to minimize the waiting period, but that also means you could already de-escalate or escalate after 3 patients if you use 3+3 design. But here, while you're waiting for data, you might already put 4th or 5th patient on the same dose level.
Right.
So yeah, that's indeed nothing comes with all good things. It's one of the drawback of rolling 6 design.
So what is the standard practice though, if you enroll patients and then you do get 1 or 2 with DLTs? Do you reduce the patients that are already started, or if they're tolerating it, do patients tend to stay on that assigned dose level? Or I guess I don't have too much experience in managing this particular design.
Usually we don't do inter-person dose escalation, de-escalation.
Okay.
So yeah, usually, of course, if you have 3 patients, you already have 2 DLT, you know right away you need to de-escalate. You don't have to waiting for data from 4th or 5th patient, you de-escalate right away. And for the 4th, 5th patient, if they're already treating on the dose levels, you might want to decrease the dose levels for, even usually one cycle, including multiple treatments, you might want to decrease them right away because you see toxicity. However, those 4th and 5th patient will not be used for the escalation, de-escalation rule anymore. I think the treatment on those patients probably going to based on physician discretion.
Yeah, that's helpful.
Okay.
And then I would also have to say in the phase 1 world, I don't know, anecdotally, there's just many investigators who feel that doses get overestimated for tolerability in accelerated titration designs.
Yeah.
And that ultimately you end up assigning too high a dose that comes down in later trials. So that's one reason why these designs are not always espoused, even though they would seem to invest far fewer patients.
I definitely agree. This is because of this aggressiveness. Of course, it's all based on probability. The chance you observe a DLT out of 1 patient, of course, you have high chance to miss a DLT. So, that leads to you overestimate MTD. Actually, this criticized also was actually for the laid down for the continuous reassessment design, Bayesian design. Because I think because it's also allow dose escalation after only 1 patient. It also potentially overestimate the MTD level. Okay, yeah. So the second class, this is the class getting popular late years. Although I think the literature, I can check back literature to 15, 20 years ago, using model-based approach for dose escalation and de-escalation designs. And for this class of design, pretty much all of them first assume or establish a dose toxicity curve prior to study initiation. Then they select the target DLT levels. So after patient get treated on the on a dose level, either 1 patient or cohort patient treated on dose levels, this model always get refitted and repeated model fitting and estimating, update the dose toxicity curve. And so the model will tell you where to assign the next patient or next patient cohort based on model predicts. So you can see, and the Bayesian ideas really here, you have a prior guess about those toxicity curve, and using data observed on the study to do the posterior estimate. Posterior means readjust or updating the dose toxicity curve. Then the curves will tell you where to put next patient cohort. So for this class, because of this idea Bayesian design, this big class of design, they all have another name that we are most familiar with. Usually we call them Bayesian adaptive design, because it's the Bayesian idea. And however, this class, I have to agree some of my physician colleagues when I discuss with them, it's conceptually and computationally complex. It's like it offers like a black box, right? We don't really see the escalation, de-escalation scale and rules. It's pretty much the model tells us. Then it feels like you don't feel like a very certain whether how aggressive or at which level the dose will de-escalate. It's just limited. So I think this is probably the reason and limit the use of model-based methods. Now, to speak of those one, I'll pick up two designs in this class and the currently popularly used. The first one is this continuous reassessment method, which gained the name for this class. usually we call them design, which we call them Bayesian adaptive design. So here, I use the figures here. So initial dose toxicity curve is specified. Usually it's called a skeleton, shown as a solid black curve here. This curve models dose toxicity relationship. Usually it's probability of DLT with increasing dose level. The y-axis is the probability of DLT, and x-axis represent the dose levels. So here, there are some technical details. I will not get into details, but just tells you here, this is the one parameter models. And if the PJ is a true DLT probability of dose level J, and this AJ is the prior guess of DLT probability at dose level J. You can see it's A1, A2, A3, the initial guess, then pretty much define the initial guess, the skeleton, the black solid curves in the last slides. Here's the alpha. The alpha is unknown parameter. It just controls the relationship between the true DLT and the initial guess. So what they do is they use this equation, this model, to link the two dose toxicity curves with its prior estimate. Then after each patient is treated, then this model get updated. Pretty much the alpha parameter. Alpha parameter will have a prior distribution, have some assumptions. Then we update, have a posterior estimate of alpha parameters. And those toxicity curve will be updated. Usually, if at current dose level, there's no DLT and no DLT observed, then the dose toxicity curve likely will be lifted up. And so the decision for next cohort likely going to be those. So if there's no DLT observed, then the dose toxicity curves will be low down, means lower chance of DLT with increasing dose level. In that sense, the dose will likely get escalated. If there are DLTs observed on current dose level, that means the dose toxicity curve will be lifted up, means potentially higher dose toxicity, higher probability of DLT with the increasing dose levels. Then the dose likely be de-escalated. Here, this is the illustration figures. See, if the orange line is the initial guess of dose toxicity curve, if for the first 3 patient cohort has no DLT, then the updated curve is likely showing here. You can see the probability getting lower, and the next dose levels, you can see if this line represent the target dose toxicity levels, then the next one, it could be go to dose level 3 or even dose level 4 here. And however, if you do observe toxicity on these dose levels, for example, you observe 2 out of 3 patients as observed dose toxicity, you can see the curves, this light blue curves gets lifted up, much higher DLT probability for a given dose level. Then if you use this target dose toxicity rate, you can find maybe dose level 2 is already, dose level 3 is all exist or exceed. Dose level 2 is potentially the next choice. So just like I'm showing here, I think this is another plot. The green means no DLT. Then this patient has green means no DLT. The curve gets lower. The red means this patient observed DLT. The curve get lifted up. Then so based on these ideas, the next dose level will be selected based on model prediction. What's the next higher dose levels that's still below your target toxicity probability, target toxicity rate. And so you continue in this matter until either the pre-specified sample size exhausted, And after you're done the trials, you have the DLT information on all patients treated on the trials. Then MTD is selected as the dose level with an estimated DLT probability closest to target DLT probability. So recall 3+3 design. The MTD is pretty much determined by the last one or two dose levels. And here, the MTD was determined by using data from all patients treated on these trials, pulling information together. I want to point out is it's closest to the target DLT probability. It really means closest could be lower or a little bit higher. Just that those levels closest, had the estimated DLT probability, the closest to target DLT probability. And so initially, the original CRM allows skipping those levels because it could be skipping the next dose level to the 2 levels higher, and also they can also allow you to add a dose level in between. And however, I think later on, because the rule, they find out this algorithm, these rules have much higher chance to overestimate MTDs. So now they forbid skipping those levels. Also add a safety stopping rule, just to minimize the chance of overdosing. Here, the idea pretty much means, based on all data we observed, if this dose level, if the estimated DLT probability of this dose is larger than the target level, it's already 95% chance, then pretty much this dose level and all higher dose level will be eliminated. So this is some technical details about the CRM designs. I think I won't get into details. So again, so they can see the idea of this design. The advantage is to improve efficiency because it use less patient to reach before they reach to MTD level, but potentially overestimate the true DLT level, as we discussed before, because it really, I think, a little bit aggressive in terms of escalation. So there are various modification of the CRM, and this one is called escalation with overdose control. It started to use two parameters to model the dose toxicity curve. And this is another one I see a lot in the protocol review from certain pharma companies. They like to use this Bayesian logistic regression model. It was two parameters, alpha and beta, to model the dose toxicity curves. And of course, you need a prior distribution assumption on alpha and beta. Then each time after you observe DLTs on the dose levels, and then you update alpha and beta to get a posterior probability, then get a the mean of posterior probability to be current estimate to help you determine the next dose levels. Here, the idea is a little bit different from the CRM because this design, the Bayesian logistic regression model, using a proper dose interval to guide those escalation de-escalations, pretty much means if you have a target dose level, for example, 25% is your target DLT probability, then maybe we think from 20% to 30% within this interval will be acceptable proper dose intervals. And this model is pretty much to find the high dose levels and to find a dose level with highest posterior probability of being of the DLT probability be in this interval. It's a little bit, yes, tongue twister. So the idea is just to using this interval to guide the selection of MTD level. Again, it also have overdose control. Also, it does not allow those skippings. So here again, this slide is probably for statisticians. You can, if you want to understand indeed how those, for example, how those model prior assumption is, how this posterior posterior probability or posterior distribution get updated, and what are the overdose controls. This is one of the tables borrowed from a publication, a paper. It tells you the models, the prior assumption that often used for those designs, and CRM, EWC, and Bayesian logistic regression model. And those are the designs we'll talk about later. So this one, I think, can help you to understand and really the technical things behind those designs. Okay, and so the next one, this new class, I myself like quite a bit. As I said before, the model-based design, for me, is a little bit too complicated for me, even for me to explain to my physician collaborators. And for that designs, it's hard to understand. Even for statisticians, we need to have sufficient knowledge. We have to use the statistical software to do the design to understand this. So it just, you know, in the clinical settings, we think the simple the better. Of course, the simple, the valid, the better. And this design, I think, was motivated by that desire. We want to be simple, but we also be efficient. So this designs, the idea is they do use model to make efficient decision making. However, it does not assume use a model to model those toxicity curve. It does not model that skeletons, and it use pretty much all model here use the interval design idea, and they so this way they can make a decision like a model-based design. So to gain efficiency. However, you will also be able to pre-specify the escalation and de-escalation rule. Can even tabulate them in the before the onset of the study. So you can see when you do the trial, you can do accordingly. I'll show the table later. Example here is based on optimal interval design and the modified toxicity probability design and keyboard designs. I think I will focus on the first one because the first one, I think the ideas makes more sense to me and performance-wise as well. So here, you know, the figures illustrate the here figure on the right pretty much shows you the ideas how of this escalation de-escalation algorithm. You start in treating the first patient or the first patient cohort at the lowest dose level. Then what you do is you really just calculate the observed DLT probability. You observed probability, then compare with this pre-specified interval for those level. This pre-specified dose interval, let's use lambda E and lambda D to specify the interval. Lambda E is a lower level, lower bound of your the interval, and lambda D is your upper bound of the intervals. So if you observe the DLT probability is lower than the lower boundary, is smaller than lower boundary of this interval, pre-specified interval, then the decision is to escalate to the next dose level. If it's observed the DLT is higher than the upper boundary of this interval, here this route, then the decision is to de-escalate the dose level. If the observed DLT fall inside these intervals, then the decision is to retain the current dose level, treat more patients on the current dose levels. So then you repeat the step until you meet the maximum sample size. So here's is pretty much a simple illustration of these trials. You treat a patient, does that meet your maximum sample size? If yes, you stop the trial and find MTD. If no, you compare with its pre-specified intervals. You know, less pretty much fall on the left side of the interval. You escalate. If fall complete on the right side, higher side of the interval, then you de-escalate. Within interval, you remain on the same dose levels here. That dose level. Actually, if you think a little bit deeper, play around, you will find a 3+3 design. Actually, it's a special case of this design. If you choose 3 patients per cohort and also use target target dose level 0.3, we can review later. Both 3+3 rolling 6 design and even the ADT, the accelerated attrition design, are special cases of this class, can be made under the special cases of this Bayesian design. Again, this is some technical details. I think I just briefly mentioned them just for the statistician audience in this statistician in this audience. So the idea of the Bayesian design here, remember, it's still a Bayesian design. You still have a prior guess and we still update that. So here is if we use PJ to denote the true toxicity rate, and of those level J, then there are 3 points hypothesis. 3 hypotheses we're trying to test and estimate. So using the phi as target dose levels, phi 1 as highest dose level and deemed subtherapeutic means, that's pretty much this dose level, and all lower dose levels are subtherapeutic, means lower than MTD. Then phi 2, the lowest dose levels deemed overtoxic, means this level and all above levels are already exceed MTD. So the 3 hypotheses, the first one is what we want is the true dose levels is, the true DLT probability of this dose level is the target dose toxicity rate. And H1 is the true toxicity rate of this dose level J is somehow below the target level phi 1 subtherapeutics, and H2 is this the true toxicity rate is above the DLT is a phi 2 is deemed overly toxic. So the idea is really just, okay, we calculate to see which one, if the conclusion is we conclude on the first hypothesis, then that means the current dose is MTD, then we should retain. The right decision is retainment. If we conclude each one is correct, then this current dose level is subtherapeutics and should be escalate. The right decision is escalation, E. Then if we conclude H2 is correct, then the current dose levels is overly toxic, and right decision is de-escalation, is D. So then incorrect escalations, incorrect decisions is R bar, E bar, and D bar. Probability of making incorrect decision is writing here. The idea of this design is to assuming, first, let's assuming the prior probability of the 3 hypotheses is the same. This is prior, this is what we assume each dose level, we assume 3 hypotheses have equal probability to be true here. Then the decision rate is to minimize this incorrect decision, the probabilities. You can calculate this lambda 1 and lambda j, and lambda 1, lambda j decides is where the toxicity interval was calculated. So lambda 1 and lambda j just here, the lambda E, lambda D here, just define the pre-specified toxicity interval. And there's some general guidance. How do you select phi 1 and phi 2 with regard to your target toxicity rate? Usually you allow 40% lower or 40% higher than target toxicity rate to have phi 1 and phi 2, which will in turn decide you pre-specified toxicity interval for the dose level. So here in can see these toxicity intervals can be pre-specified. Remember, for the 3+3 design, rolling 6 designs, the target toxicity rate is already fixed. You can only have one. That's only one choice. Here, you can free to choose a flexible choice of any DLT rate. If you choose 0.3, that's usually where we choose. That's mimic 3+3 designs. Then the toxicity interval, lambda E, lambda D can be specified, can be calculated here. That's the toxicity intervals for target DLT probability of 0.3. Here, based on these rules, based on these intervals, the escalation, those escalation and de-escalation boundaries can be specified, can be clearly calculated and tabulated as shown here. Because right, we know observed DLT, number of DLTs, which number of DLT will give us numbers within and above this intervals. So this one, the number of can tabulate the escalation de-escalation rule. Based on this table, you can see we do not require a fixed cohort size. You can take, for each dose level, you can treat any numbers. We can take clinician inputs. Maybe initially we want to treat a single dose cohort and switch to cohort 3 once a concerning toxicity occurred. Again, this idea of ATD. Also, escalation and de-escalation can happen at any time. There's a continued number of patients treated. At any place, you can decide to escalate and de-escalate. This can handle some passive cohort change, as I mentioned before. For example, you plan 6 patients at the study, accrue 6 patients. However, only find 5 patients eligible. But still, for this one, for 3+3, you probably have to treat 1 more patient on dose level in order to make a decision. And here, 5 patient escalation de-escalation rule are specified. So you can always escalate and de-escalate. And last part is different from a rule-based method, because you can see those are very attractive features because it's really clear and straightforward. And the last part is when the DMT was determined, it really pulls information from all those levels. The idea is used as isotonic regression to estimate toxicity probability of all of those levels and select MTD dose level for which the estimate of DLT rate is closest to the target. Again, the similar idea, closest could be higher or lower or higher, just based on this regression. The interesting part is because escalation and the MT determination are 2 components of this of the design. Once you've done the buy-in designs, you have a DLT information on all patients from all those levels. You can use different methods to estimate MTD. You can feed the data to the reassessment model, and you can use that one to estimate MTD as well, just some flexibilities. So for these designs, I think they have a few attractive features. First, you use observed DLT, which to make a decision. So make this design transparent and accessible to non-statisticians. And also because it's using the observed DLT, it guarantees de-escalation when those level, when the observed DLT rate is higher than some de-escalation boundaries. You know clearly is once you reach this level, you will de-escalate. So it's kind of you feel safe because to have this de-escalation guarantees. Also, this one, although you use this idea of Bayesian ideas, but we do not have to update the posterior distribution, and we do not have to update the models. And the idea is already implemented in these tables. This is how we're gonna do things. And so I think those features, for me, like it's pretty attractive. And also when I explain to my physician collaborators, they understand the ideas and it's easy to implement. This is a table you can do accordingly when you do escalation, de-escalation.
Fangxing, I have a quick question.
Oh, yeah, sure, go ahead.
You mentioned that this will be repeated until the maximum sample size is reached. How do you determine the maximum sample size?
That's a very good question. I think usually, of course, the selection of sample size is always a big question. You don't want to, or if you have 5 dose levels, you probably don't want to just only allow 10 patients. And you have I think my understanding of this is first of all, you have reasonable. For example, I allocate 25 patients for those escalations. Also, the trial may be able to, I can also design a range, for example, 20 to 30 patients. For the models, if after 20 patients, I already put 6 or 7 patients on the same dose levels, it keeps telling me that's MTD levels. And I think I can safely choose that level as MTD. I do not have to put more patients on the study. So I think this is it's two sides. You give a reasonable sample size range, and you also can take feedback from the model. Does that make sense?
Yeah, thank you. And there are a couple of questions in the chat. I'm not sure if you want to take a look now or at the end.
Oh, okay. Oh, thank you. I didn't realize the question come in chat. Oh, the first question is the CRM. The continuous reassessment model example has cohort size of 3. Does the cohort size has to be fixed in advance and across all those levels? The answer is no. You do not have to fix cohort size. And I think the CRM initially when they were designed, it provides flexibilities. You can choose just like the binder here. You can choose one patient at one patient cohort initially. You can choose whatever take physician input, or you can start with 3 if physician thinks that the starting dose level is already have some data showing to be closer to MTD, close to the MTD. So the answer is no. You do not have to fix cohort size, and you do not have to have the same cohort size across those levels. And the second question is, any consideration in terms of the window of DLT observation in the IO setting, as we know that some RRAEs occur really late? Yeah, this is another question. I think I think at the end of talk I mentioned one thing, and this is delay toxicity. This is one of the issues it's important, but I did not cover in this study because the delay toxicity, how this affects trial conducting. Of course, you can always extended DLT observation period, but how that will affect designs, I think this part, this is not an easy answers. This talk does not cover if you have a delayed toxicity, then what will be possible or efficient modification of the designs we'll talk about here. Nice to see you, Jeff. How are you?
Good. How you doing?
Good.
Who is that? Who is that? Is that? I'm sorry.
Fangxin?
Yeah. I was just actually sending you. It's actually online, but I can pull the slides. We were in a phase 1 study of a Novartis agent, AUI 922, and had it been 3+3, it would have stopped at 36 mg per meter squared, and by CRM was 70 mg per meter squared, and the 70 actually ultimately had activity in lung cancer. So there are some very good examples of where 3+3 fails, where it's too conservative. And then I guess in CRM and in Bowen, you can go up and down. In other words, if CRM sends you back down, you can still go back up, right? And the same thing in Bowen. I think that's what people really like about it. So if you end up with two DLTs and 3+3, you're done. Whereas in these other designs, you can go down, but then if you enroll more patients and you're okay, you can go back up. So people really like that. Bowen is now the rage. Nearly all the NCI CTEP studies, a lot of them are using Bowen. This has just become the new thing that's in vogue.
Yeah, I think I had the impression we had an LOI with Michael. We used a Bowen design too last year. Yeah,
that's right.
Yeah. I agree. I think a Bowen, I like it because I think it's simple. The idea is simple and it's really easy to implement. It has the advantage of what you described. You can go up, you can go down and go up again. So it overcomes the potential too conservativeness of 3+3. likely
Yeah.
to get to the MTD, get much closer to the MTD, the true MTD level.
Right. I
Yeah,
think that's been the major, but the Bowen is definitely a lot easier for people to grasp. So I fully agree with everything you're saying. I think that's why it's been popular, because people kind of, like you say, there's a transparency there, and
Yes.
people like a bit of the rule component of it.
Yes.
So,
You know, I
and
think
it's
nobody
also
like,
doesn't require as constant a statistician vigilance, because you don't have to remodel every minute, right?
exactly. Yeah.
Yeah.
Yeah, exactly. You know, of course, close collaboration is always important. However, you know, this gives you freedoms of flexibility. You know what to do based on the table. Compared with the CIM, you constantly have to update models. Sometimes the model get complicated, and we know the performance of CIM heavily depends on how good your initial model is.
Right.
If your initial model is wrong, you know, if those curve you assume this way, it actually this way,
Yeah.
yeah, and then
Right.
then you will be actually the MTD will be way off. So I think we like model, but we also try to be less parametric, because this way is more robust. Okay, I think we're back. So this is, I got a few more questions. Okay, this is the one question said, if you have too few dose levels, does the regression model have trouble to estimate the MTD? I think my short answer for this one, if you have a too few dose levels, like less than 3 to 4, 3 plus 3 is perfect efficient compared with any other dose level, any other designs. I would suggest you just go with 3 plus 3. And you will imagine if you have only like a 2 or 3 dose levels, and you probably don't have a very good idea regarding those toxicity curve, right? You don't, because it's really 2 or those levels. So for this question, I think a 3 plus 3 is still the way to go. Okay, the next question is, it's from Jeff. There's a very good example of a Novartis agent, AUY-92, that could have stopped at 36 milligram per meter square by 3 plus 3 and ended up at 70 milligram square meter by CIM. At those levels that ultimately have activity in subset of lung cancer. Yeah, presuming that we can up and down with either CRM and Bion. Yes, that's the advantage of CRM Bion is what I think one attraction is it can go up and down. When you accumulate more data, if you see 1 in 3, then 2 in 6, you stop by 3 plus 3. And in Bion and CIM, you give the dose level one more chance. So you can treating for 7th patient, no toxicity, 8 up to 12. So that does give you more chances. But again, I have to see some simulation have shown because of this feature, nothing comes with all just disadvantage. Some simulation has been shown on certain scenarios, they could also overestimate MTD, have higher chance to overestimate MTD.
Yeah. No,
Okay.
thanks. Okay.
I think this is from BI again. I mean, if you have very few dose levels after escalation ends, I see. So I think this question really is, if initially we have like a 5, 6, 7 dose plan, however, the CIM or stops after the escalation. I mean, after 2 doses, you stop escalation. I think the answers, because we're based on model, the models, like we discussed, the CIM the performance of CIM heavily depends on the initial dose toxicity curve, how close to the truth, and we use observed data to update, to modify, to use posterior probability estimate of the curves to getting closer to the truth. So imagine if you only have 2 dose level tested, and then you use only those data to update the toxicity profile, those toxicity profiles. Then you observe data is really limited. Means you have the posterior probability still dominated by the prior assumption. So I think this way, this is probably even more depend on the prior guess of those toxicity curve. So I think the model fitting probably gonna be a little bit problematic in that setting. No more question in chat. Let's continue. Yeah. Thanks, Jeff, because he mentioned some of the things I forgot to mention for the Bayern and CIM designs, because of the features go up and go down and go up back again. And thanks, that part I missed. So I think I won't get into details about this technical of this modified toxicity probability interval designs. This idea is really calculate the posterior probability of 3 intervals under dosing, right dosing and overdosing. Which one has the highest probability? Then you make a decision of that. So I think I will skip those 2 parts of the technical details. If you want, we can always discuss after the training session. Again, this is another thing I talked before. I think this will be useful for the statisticians to understand deeper regarding technical details and assumptions and some overdose control rules for each design. Okay, and this is the online resources I like to summarize here, because like we said, the CIM is like a black box, usually provide operation characteristics by simulations, but those they all already have software freely available. So here, this is the website I include in my slides, and this is the phase 1 design, and this is the CIM, and this is a Bion suite, and this is a keyboard design, and even for Bions, they are actually a lot more than I initially got to know, single agent combination and things. You can play around with those, and it tells you you can either use a web-based software or you download to your computer. For example, let's go to a CIM. This is pretty much the input. What's your target toxicity rate? How many dose levels you have? What about cohort size? I said. Although for this one, I have to say, because this is a software pre-made, it takes a fixed cohort size for those levels. But this is just, but the actual model can be more flexible. But here is, has to be simplified to provide a user-friendly software. And this is the skeleton, the initial dose toxicity curve, and this is a scenarios. What's the true DLT levels of each dose levels? And you can calculate, for example, here, the operation characteristics using simulations here. It's a one scenario. You can provide multiple scenario just to see what's the true DLT rate, what's the selection probability using this design, how the chance you choose which dose as your DLT. So this is the one. Okay, now let me go back to my slides. Okay, so that's the I think if you want to, you can play around with those one together with some publications will help you to actually understand, then use those tools to actually design your studies. And also for the CIM, this is based on logistic regression model, has R package. Our package Bions, as we showed here, have the user interface window based, and also shining online apps, and also R package. MTPI keyboard both have R package, so you don't have to actually write your simulation or write your design. You can use a package, but still, I would suggest you understand the method, you understand the ideas, and some necessary technical details before you're using those tools, so you can actually understand which parameter means what, and you can interpret your operation characteristics. Okay, next. So you can see, I think we don't have time to cover all of them, but we still cover quite a bit of phase 1 designs. Now we know we have many options. We always say it's good to have options, but sometimes it's not it's not when when you have too many options, then you have a problem too. The problem is, when do we use which, right? There's so many designs. The short answer is no one size fits all. There are definitely is. I will not recommend. I don't think anybody can recommend any design that fits all designs, all purpose. So rather, you want to find some specific method, then some specific method performs better or more fit for purpose for particular trial. So you really just, based on your study, based on your objective, and based on your feasibility, how many patients you can afford and things, then you choose the one that fits better or performs better. Those are slides, I keep them. This I use initially when I give this talk at ASH. And there are some possible considerations. And then those considerations were proposed by using statistical simulation on, comparing various designs on the different scenarios. There are some general considerations. For example, how wide is your therapeutic window? Is the therapeutic window narrow or wide? And how much prior clinical experience you have? Because both of those two factors will determine how many dose levels you have, right? For example, we know most novel agents test first on a solid tumor. When you move to hematological disorders, we already have pretty good ideas regarding what MTD gonna be. So, and also, is adult trial or pediatric study? Most of the agents, again, also first test in adult trials, then move to pediatric studies. When in pediatric studies, usually we start with 50%, 75% of MTD of adult, determining adult trials. So we have pretty good guesses regarding the MTD. That means we're gonna have a much fewer, probably only a few those levels. Because, like I said before, it really depends on how many candidate dose levels we have in the study. If you only have a few, less than 3 or less, I would suggest 3+3 design is still a way to go, because it's perfectly efficient and we know everything about it. And if it's more than a few dose levels, it's like more than 6 or 8 dose levels, I think the CIM and the buy-in are the way to go, because it's because of the efficiency in terms of dose escalation and thing. And if something in between, I think it's less clear, but based on the conversation Jeff and I had, I think the buy-in will probably the way to go is the combination of efficiency and this, the straightforward implementation. And other consideration is, again, is really how good knowledge on the true dose toxicity curve. This is really not only about CRM, because the CRM heavily depends on your initial guess about those toxicity curve. Also, you want to know whether those toxicity curve is gradually increasing. The probability of DLT gradually increasing with those levels, toxicity level with those levels. Then you probably don't want to use 3+. You want to use some other designs that 3+3 or CIM. And those handles like jump in toxicity between adjacent levels better, just like here. And if you have a fairly constant or safe toxicity profiles, maybe for some molecular target agent, you don't have fairly constant and safe, you determine already pretty safe, then a CRM and buy-in might be the way to go. Of course, in these settings, maybe toxicity is not your major concern. You really want to maybe other endpoint, maybe the PK levels, that will be something not covered by this training. Okay, so I guess I really just, my suggestion for the physician audience in this training session, then my suggestion to you is collaborate with a trained statistician. For statistician like me, like us, we have to gather enough knowledge. We have to keep train ourselves, know those methods, the assumptions, the operations, and the pros and cons, all the things, and in order to actually use those designs. So I would really suggest you to collaborate and with the statisticians, they can ask you questions, translate your questions or your goals into statistical language, and give you some ideas and explain to you and what will be a right designs, what will be your concerns. So again, collaboration is the key. And there's some things I like, some I got a question in the audience I didn't cover in this talk. What if like new agent, molecular target agent, immunotherapy, a delayed or chronic toxicity? How do we do design to monitor, to handle those delayed or chronic toxicity? This is not, I did not cover. I just pretty much using a canonical one cycle observation of DLT as underlying situation for this talk. And what about combination therapy, right? A single agent toxicity, and you know the single agent those toxicity curve. How do you model interaction between the two agent? And second, and the last one is what if like the DLT toxicity is not the appropriate endpoint for phase 1? Is you really care about the optimal biological dose, then maybe pharmacologic guided dose escalation should be used instead of using toxicity guided dose escalation. Also, I did not cover anything about intra-patient dose escalations, and so you can see phase 1 design can be really complicated. And let me see. Okay, next I want to briefly cover those expansion cohorts, because those expansion cohort is a important component, maybe become more important lately. So we know the expansion cohort traditionally is 12 to 15 patients treated on the MTD level to further characterize those toxicity profile, and also additional assessment of the MTD, preliminary assessment of efficacy. However, in the recent years, the expansion cohort has been given many more tasks than what we listed here. I copied this from the FDA draft guideline for expansion cohorts, and you can see here, expansion cohort, the multitask expansion cohort can be used to confirming safety and all recommended phase 2 dose level, evaluating preliminary efficacy, and evaluating PK and PD aspects, and biomarker development, and evaluating more than one therapeutic drugs. So it really go beyond the traditional or the conventional dose is a small scale dose expansion cohort and in the phase 1 trial. So and this is the multi expansion cohort trials usually, and it has more than 3 or more additional cohorts. They expansion cohorts, each of them have a cohort specific objective. Usually it's typical of a phase 2 trials, means expansion cohorts have a phase 2 trial, phase 2 objective. For example, assessing anti-tumor activity in a disease-specific setting, specific population. Of course, the goal of using multiple expansion cohort is to expedite drug development. And nothing comes with all advantages. The challenge is, and it's timely dissemination of new safety information, because it's critical that investigators, RRBs, and regulators are updated with the new safety information so they can provide necessary oversight for protection of human subjects. So the investigator can ensure the patient can provide adequate information for consent. And also, because you do a large-scale expansion cohort, it potentially can expose a large number of patients to potentially suboptimal or toxic dose levels, because MTD hasn't been further verified in the expansion cohort yet. And sometimes you do unplanned analysis, you could provide mislead interpretation, actually can further slow down the development. So it's really, I think the key, one of the suggestions in the draft guideline is imperative that sponsors, means usually pharma company, establish an infrastructure to streamline trial logistics, facilitate data collection, and incorporate plans to rapidly assess emerging data in real time and disseminate interim analysis to investigators. And so you really have to have the necessary infrastructure to handle the challenging brought by this multi-expansion cohort study. I want to give you this one example. This is one example about the development of nivolumab, a PD, immunome checkpoint blockade therapies, a paper published on New England Journal of Medicine. So in this phase 1 design, phase 1 study, it has MTD expansion in 5 disease cohorts. Non-small cell lung cancer, advanced stage melanoma, 5 disease cohort. MTD expansion means they determine MTD, then treat a patient on the MTD level. They also have non-MTD expansion in 3 additional cohorts. And later on, 2 of those additional cohorts was further amended to become a randomized 2-arm study to comparing 2 dosing levels. You can see in the melanoma, by cohort randomly assigned to 2 dose levels and lung cancer 2. So end up this phase 1 study has a total of 14 expansion cohorts and ad hoc designs involving 5 amendments. You can see those expansion cohorts were not driven by safety, which is the goal of phase 1, but driven by the initial efficacy signals. So making them actually phase 2 studies. So this phase 1 study has successful outcomes. However, because it's ad hoc designs to change later, also with issues like increased risk of false positive identified anti-tumor effects, because we test so many hypotheses in so many disease areas, increase what we call top 1 errors. And so I think for the expansion cohort, after based on my own experience and reading the guidelines and reading the trials publication, I think those expansion cohorts are here to stay. So of course they have to have a scientific justified cohort specific objectives, then an appropriate design for the specific objectives. For example, if you use a phase 2 sample size for the expansion cohort, then it has to have a phase 2 objective and phase 2 designs, sample size justification, monitoring rules, analysis plans. Just like we always said, if it's quack like a duck or work like a duck, it has to be a duck. So, of course, the advantage is we can move after this kind of those expansion cohort, we can move directly to randomized phase 2 or phase 3 designs. And the idea is really just to replace early phase 2 trials, can get to the to put the drug into the market more quickly. So after actually when I prepare the trainings, after talking about multiple expansion cohort, and I recall in recent years when I review protocols and design my own studies, we also use this multiple escalation cohorts as well. Means the escalation cohort is not in, it can be parallel multiple escalation cohorts. So I give you 2 examples just to illustrate my points. And so this one, the first one is the phase 1 study and study the combinations of 3 drugs using a rolling 6 designs. So in the latest amendment last year's, they amend the protocol to add additional combination of the 3 drug combinations. So the idea is for these combinations, it's still using rolling 6 design. However, the starting dose of this one, the main drug, have a CC-lib. The starting dose will be the same as MTD determined from the combination in the first combination. So the starting dose of palbociclib is the MTD determined from this combination. So it means it's already been done. This escalation already been done. And it only allowed de-escalation. So it means If this MTD is, this MTD level is fine, then phase 1 trial is done. Phase 1 escalation is done. If not, we only allow you to go down, not going up. So you can see that this modified escalation ideas is really sequential development of two combinations. Of course, each of them followed by expansion and also 7-2-stage designs for the expansion cohort. And this is another example. Study the same combination. This is the same agent, but in different disease cohort. 2 escalation cohort. Arm A is for relapsed refractory non-Hodgkin lymphoma and multiple myeloma. Arm B is used AML. So the same drug is the study of this combination drugs, but in 2 different disease cohort. The reason because prior data suggests different dose toxicity curves in different disease, different disease area. Lymphoma and leukemia very different. And this design they use the Bayesian hierarchical logistic regression models and using different initial guess about those toxicity curves and do parallel independent dose escalations. At the end of the day, they could select a common or separate MTDs for different arms. They even says, okay, if the data suggests non-Hodgkin lymphoma and multiple myeloma shows difference in those toxicity curves, and they will further divide arm A into 2 parallel independent dose escalation cohort. Again, of course, for this protocol, it also has a full expansion cohort just to individually, independently evaluate preliminary dose efficacy data in different disease areas. I quickly cover the phase 1 design about also the expansion and escalation, multiple expansion cohorts. So we can see phase 1 trials and designs could be very complex. It's definitely not straightforward. Maybe some are, but not many could be very complex. So also, like I said, my goal is to provide an array, let you be aware the array of designs approaches that have been developed for conducting flexible and efficient trial designs. So the important thing is, you know them, you have options, you know that they are possible candidates, then you can select between them when you have your own problems. Of course, there's no one size fits all. There are always one or more fits for the purpose. That's why important things is that statisticians and clinicians, they have sufficient knowledge and about the novel design, and they can be comfortable and confident to use them. Then they talk and collaborate. So lastly, I want to reiterate is communication and collaboration. In it's in this healthcare settings. I think everybody knows that many stories shows the importance of collaboration and communication, and can speed up the whole drug development process. I think that's pretty much all my trainings, and we have some time for questions if anybody has. Let me see, there's one question in the chat. Oh, this question is about related to what asked before. If we have, if initially we have more than a few dose levels, for example, 6, 8. However, the trial stop, you have only a few dose level after escalation ends. How about estimating MTD using the Bayesian? That's a good question. I don't have a very, answer for that, because in terms of that particular scenarios, like I claim, I do not know the techniques and details about all the designs, but I can tell you what I think is, regardless, because the Bayesian use isotomic regression to estimate probability of DLT across those levels, I think it will be a little bit problematic if you have only a few dose level actually be tested, means you have observed the DLT level on only a few dose levels. It will be a problematic to estimate MTDs. how robust, how much problem there will be. I think we can search some literature and some simulations might have already done on this topic. Just I don't have the answer now. And I want to thank everybody again for coming to the sessions. If you have any questions, you can feel free to contact me. I'll be happy to discuss with you regardless of technical details or some practical issues then after this training session.
Thanks, Fangxin. If everyone will give her the Zoom reaction or unmute and clap. Thank you.
Thanks, everybody.
Thank you.
Have a good afternoon.
Thank you.
Thanks,
Thank
Fangxin.
you. Thank you very much. That was terrific.
Thank you. Thanks for coming.
Right. I know.
Bye, Jeff. Bye, Emmanuel. Bye, everybody.